So particularly for microRNA identification, if we talk about microRNA identification from the genome and transcriptome or degradome, these are based upon the pipeline. Several pipelines are there, and the specific tools for microRNA identification are also based on some languages that are used in Linux system. So I have just prepared the table because we all are having simple laptops and we don't have Linux system. So that's why I have just prepared a table of so that we can just go through it. What are the tools and what are the languages that are used for a particular tool, and what are the systems. Okay? So these are the major tools which are used for microRNA identification. Like first is miRNA, and in this table I have also added the year of the formation of that particular database or tool and reference. Then what are the platforms that are required for that particular database? What are the features of, like miRNA has only microRNA prediction feature, and it is based upon Perl, Python and R language. Anyone is familiar with languages? Which languages? So you know about this, that these tools are specifically based on Perl, Python and R and Java, C++. So these tools are basically languages based, like miRPlant, miRDP2. If I talk about our lab, my super senior used this tool, miRDP2 tool, using Perl language, and so these are the main tools. One is miRPlant, which is based on Linux, which requires the platform Linux, Windows, Mac OS. Then it is also used for microRNA, only used for microRNA identification, and it requires Java language. So then miRExpress platforms are Linux and Mac OS. In miRExpress, as the name indicates, it will also give the expression of the microRNA. Then miRKwood, it requires Linux, Mac OS, web-based tools, and features are microRNA identification, microRNA prediction. It is based on Perl language or C language, and specifically for plants. Then miRDP2, it requires Linux, Mac OS, and microRNA is similar to the previous one, and it is based on Perl language, and it is used for animals. So this is just a table to show that these tools are there for particular identification of microRNA. And the pipeline used by my senior for microRNA identification, I have also added this. So this is the pipeline that have been used by my senior. First is small RNA sequencing. Small RNA sequencing is different from the total sequencing, like Illumina, jo transcriptomic sequencing, it is different from total transcriptomic sequencing, because total transcriptome includes mRNA, larger RNA also. But in small RNA sequencing, only small regulatory RNAs like microRNA, siRNA, pvRNA, lncRNA, these are included in small RNA sequencing. So basically, first if I talk about from the basic, she have isolated the RNA, total RNA of the plant. She have worked on chlorophytum, and she have isolated total RNA of the plant, and from that total RNA, by using specific kits, she have isolated small RNA. After checking the purification of that small RNA, she have sent the samples for sequencing, and these sequencing data she have analyzed through this pipeline. from the basic. So we are starting from small RNA sequencing, and after that she have done read quality check, the reads of the base pairs. She have checked the quality like based on the Q score. Then after the quality check, the reads with the better quality are filtered, and then adapter removal. Adapters are basically used for sequencing. These are specific primers which are used during sequencing so that they enhances the sequencing. So these are the adapters. The removal of adapter is also important because that is not the part of your sequences, original sequences. Then after quality check, adapter removal. Now after adapter removal and quality check, you have all the small RNAs like tRNA, rRNA, then microRNA, siRNA. Then she removed all other non-coding RNAs based upon different properties, like tRNA have different properties. So using that property, she have removed all the RNAs step by step. Then she have left only with microRNA. Now to check the authentication of these microRNA, she have also identified these microRNA or align these microRNA with miR-base microRNA. Like she have identified from chlorophytum, and parallelly she downloaded the microRNA of chlorophytum from miR-base, and then she aligned both. Like 180 microRNA are present in miR-base, but she have 250 reads. So out of 180, all are aligning with the 180. It means 70 microRNA she have identified are novel. So this she have done, and then further she have also characterized these novel RNAs and also submit these novel RNAs to the database. This is the basic study about microRNA identification. Is it clear? Then after microRNA identification, she have done target prediction, because microRNA only function through its target. There is no direct function, it only functions through its target gene. Then after target prediction, the genes which are the targets of these particular microRNAs, these genes are further analyzed using gene ontology and KEGG pathway analysis. Basically KEGG pathway analysis give all kind of pathway like hormonal pathways. It will highlight your genes which are present at any step of any hormone pathway. So this is the basic study for microRNA identification she have done. It is this work is already published. Now today we will cover a major authenticated tool for target prediction of microRNA. There are several tools, but we will cover psRNA target today, which will give the most authenticated targets of microRNAs. You can, first we will cover the basics of the tool, then we will also do hands-on on it. So psRNA target is plant small RNA target analysis server. Basically it is which features two important analysis functions. There are two main features of this tool. First is reverse complementarity matching between small RNA and target transcript. It will give the results according to the complementarity of the microRNA and its target, because microRNA has to bind to its target. If it want to either do translational repression or degradation, then it has to do show the complementarity with its target sequence. Then only it will bind it and degrade it. Is it clear or not? So first is reverse complementarity between small RNA and target transcript using a proven scoring scheme. And next is target site accessibility evaluation by calculating unpaired energy. Like there is a target of microRNA, and that microRNA has 100% complementarity with its target. But that target is not accessible to that microRNA. Then it will not bind to the target. Target accessibility is also important for binding of the microRNA to the target. Instead of complementarity of the sequence, accessibility is also, both things are important, complementarity as well as accessibility of the target. Clear? This point is clear or not? Yes. Instead of complementarity, accessibility. Just suppose microRNA is 100% complement to its target, but the target is not, it form loop or it is busy somewhere else, so it is not accessible to the microRNA. Then what does complementarity do? Like, so target site accessibility evaluation is done here in this tool by unpaired energy. The more is the energy required for unpairing of the target, the lesser are the chances of the microRNA target binding. जितनी एनर्जी ज़्यादा चाहिए होगी अनपेयर करने के लिए, उतनी ही माइक्रोआरएनए जो है वो कम असेसिबल हो पाएगा. जितनी कम एनर्जी में वो टारगेट अनपेयर हो जाएगा, लाइक टारगेट ऐसे पड़ा है, अगर टारगेट जल्दी उसको कम एनर्जी चाहिए अनपेयर होने के लिए, देन इट इज इजीली असेसिबल एंड इट विल 100%, it will get 100% binding to the microRNA. So lesser is the unpaired energy, more are the chances of the complementarity and binding of the microRNA to its target. Is this point clear? So by calculating unpaired energy required to open the secondary structure around small RNA target site on mRNA. The psRNA target incorporates recent discoveries in plant microRNA target recognition. It distinguishes translational and post-transcriptional inhibition, and it reports the number of small RNA target site pairs that may affect small RNA binding activity to its target transcript. Basically, it also tell about the whether that microRNA is inhibiting the target translationally or it degrading it, it will degrade it. It also signifies that. You will get it clear when we will do hands-on. Now there are some parameters of the psRNA target. We can also use default parameters, and we can also change parameters according to our need. So first parameter is complementarity. psRNA target evaluates complementarity between small RNA, and this is clear from the previous slide also, that it evaluates the complementarity of the microRNA and target. Then is multiplicity of target site. This is very important point. Multiplicity means that sometime a microRNA can cut or can target that specific site more than one time. Like microRNA is binding to the target, it is not compulsory that it will only cut on one site, it can cut on two sites. So this is the multiplicity of target sites. This tool is also providing these details about the microRNA and target. So it has capability of returning multiple alignments for each small RNA target transcript pair. Now these multiple alignments of the microRNA and target site can lead to the formation of transacting small interfering RNA. I have also taken the name in the previous lecture, previous tutorial about transacting small interfering RNA. Have you all understood about, familiar with transacting small interfering RNA. So, returning multiple optical alignments enable the reporting of multiple target sites for each small RNA target transcript pair. This so-called multiplicity of target site is especially relevant to the biogenesis of small interfering RNA, because existence of dual target sites of microRNA on specific target transcript has been reported to be an effective trigger of transacting small interfering RNA biogenesis. Then is target size accessibility that we have covered in previous slide. The psRNA target server analyzes the target accessibility using RNA UP program. These programs, RNA UP program, these are just running at the back-end pipeline of the tool, and this tool is very well available on the Google and we can use it easily. And these programs, these features, parameters, we just set the parameters, and by using back-end pipeline, the tool will give you only the results. In VINA package, RNA UP calculate the UPE unpaired energy, which is energy required to open the secondary structure of the target. Less energy represents higher possibility of an effective target site because the secondary structure may prevent small RNA and target site from contacting. This is simple. Then is translational inhibition. In plants, it has been observed that mismatches occur around the center of microRNA mRNA complementarity region. This is not compulsory that a particular RNA can cut the target site only if matches, if it's shown the complementarity to the 100%. There is a seed sequence of the microRNA that is a basically 7 base pair haptameric sequence in the microRNA. MicroRNA is basically 19 to 25 nucleotide, like, right? And there is a haptameric sequence in between of the microRNA, that is 7 base pair sequence, which is compulsory for the which is compulsory sequence if that microRNA has to target some gene. So that haptameric sequence is very important, that it show the complementarity with the target, and other base pairs may or may not show the complementarity to some percentage. It means that seed sequence complementarity to the target is very important. Clear this point? So in plants, it has been observed that several mismatches can also occur in the around the center of microRNA mRNA complementarity region, tend to disable the cleavage activity of the RNA induced silencing complex. It means due to these mismatches, that microRNA can not will not be able to cleave the target. But however, the binding of microRNA to the RISC still can block the gene expression at translational level. Then it will do translational repression. This point is also clear. The psRNA target server reports translational inhibition potential when a mismatch is detected in the central complementarity region of the small RNA sequence. So this is the input. We will start hands-on. You can just search psRNA target on the Google. Have you all got the PU Wi-Fi net? फ़ोन से कर लेती हूँ मैं भी। मैम पीयू का नेट चाहिए था मुझे। हाँ भेज दो ठीक। हाँ ये मैम ने भेज दी मुझे। So have you all opened psRNA target? Have you opened this page? Then open it. Have you all opened this page? First bench. psRNA target. अच्छा थोड़ा याद देखने दो।
इसका काम हो गया है ना?
नहीं दोबारा खोल लूँगी। Have you opened this page of psRNA target? So here as you can see there are three tabs. First is submit small RNAs. In this tab we just have to submit small RNAs like microRNA, and cDNA library is already present there of different species. There are some files you have downloaded today. क्या
आप सबको मेल आया होगा ना, वो फाइल्स डाउनलोड।
files? There are three files. Have you downloaded these files? So first one file of them is microRNA, miRNA file. So you just have to choose that file and upload here. Basically that is the file of microRNA of grapes. So you have to choose grapes here, Vitis vinifera. Here. You can choose any grape. Prefer wine grape, vinifera. Have you all uploaded that file of microRNA and choose the vitis vinifera? Now come downwards. So these are some parameters. We can also use default parameters and we can set these parameters. Like first is, this is of top targets. Top targets means like one microRNA and that genome cDNA library contains more than 200 targets. These microRNA of grapes can have more than 200 targets in the genome of grape, but it will show 200 top targets which are authenticated targets. This is for that. Then is penalty for GU pair. Have you heard about wobble base pairing? So this is the penalty for GU pair of microRNA and target. This is 0.5. These parameters are just we have to set, and this tool will use these parameters at the back end pipeline and give only results. Then is expectation. Expectation is 5 here. Expectation energy is basically the expectation of that microRNA that it will target the particular gene. Then is penalty for other mismatches, because this is not a compulsion that microRNA can only target if it has 100%. It can have some mismatches also, but provided these mismatches can be diluted in the pipeline based upon these parameters. Then extra weight in seed region. Seed region of microRNA is basically the central region of 7 base pairs, which is very important for the complementarity. And extra weight in seed region means that sometime in the seed region also 1 or 2 base pair, 1 to 1.5, basically 1 base pair can also show mismatches. Seed region is from 2nd base pair to 13 base pair. We have just added, these are the default parameters according to the tool. Then mismatches allowed in seed region, this is 2. HSP size. HSP size is the size of microRNA. Minimum size of microRNA is 19 base pair. Then it can also allow bulges, gaps. These parameters are set basically that it can give maximum to maximum results, max to max data. Then penalty for opening the gap. There are some gaps between microRNA and target, and this is 2. Penalty for extending the gap is 0.5. Translational inhibition range is from 10th nucleotide to 11th. If it is binding, it is showing the complementarity to 10 and 11th nucleotide, but however it is also showing mismatches to other regions, because as we have covered that translational repression only occur if it shows mismatches to the other regions, and if it shows full 100% complementarity or 80% complementarity, then it will cleave the site. But if it will have mismatches, then it will show translational repression at least. So then upload and submit. You can click on upload and submit. मैम मैं अपनी मेल खोल के इसमें से फाइल्स ले, इसमें हैं फाइल्स, बोली नहीं, इसमें भी चाहिए होंगी. मेल खोल लेती हूँ ना अपनी, तो सर की मेल खुली हुई है. Have you all got the results, any of you, after upload and submit? Okay. ठीक है. Have you got the results? So these are the results of the target prediction of microRNA of grapes. So this is showing the microRNA. As you can click, it will give the accession and the sequence of the microRNA. Then this is the target accession, target gene basically, which is the target of the microRNA. Then is expectation value, then is unpaired energy, then this tool is also giving the alignment details, that this microRNA is almost is 100% aligning with the target from base pair 652 to 671 of the target. Then multiplicity is 1, it can only cut from one side, and inhibition, that it will cleave, because as we all know it is showing 100% complementary, that's why it will cleave the target. So these are the results. Multiple miRNA? Target accession or miRNA? Because there are different miRNAs which are targeting different genes. No, the file contains whole full miRNAs, 186 miRNA of grapes, and these are different miRNAs. You can see in the file also. These are all different miRNAs of the grapes. This file contains, this file basically I have downloaded from miRBase, as we have covered yesterday. I have downloaded this file from miRBase, and this file then in the FASTA format I have used for target prediction. Unpaired energy is important, but it is not available in the results. I don't know why they have not updated this. So these are the results of psRNA target. Is it clear? So by just clicking on the target accession, you can get the sequence of the target also, and you can just using Ensemble, NCBI, PubMed, anything, you can just get the details of this gene, that a particular microRNA is targeting this gene, and we can immediately get the details of this gene, because this tool is also giving the sequence of the gene. Have you all opened the sequence of the gene? So this part is clear. Next is, so here you can, in the other tab, you can submit target candidates. You can open this tab. Have you opened this tab? So in this tab, you just have to select the microRNA from the particular species, and here you have to add the candidates. So I have also provided to you the file of the genes with name CDS new. You can just paste the sequences or upload the file, anything. So these are the BTB genes of the grapes. So here also you have to select the microRNA of the grapes, which is vinifera. Have you all selected both of these, uploaded or selected? हो गया? So these are the same parameters. Now upload and submit. Have you got the results? Have you got the results? Have you got the results? Okay. So these are the results of BV-BTB genes. From here, from this tab, you can just predict basically the targets of the particular gene family. So what are the miRNAs that are targeting a particular gene family? Because the file you have contain only BV-BTB family. So from this tab, you can just calculate the number of the microRNA or the family members of the microRNA which are targeting the grape BTB family. So here VV microRNA 845C is targeting the BV-BTB29 gene. From here also you can just download the sequence of the gene or microRNA also. So same expectation is 2.5 unpaired energy they have not uploaded. This is showing the alignment. As you can see here that alignment of the seed region, the haptameric sequence which is present in between of the microRNA is complete, but the alignment from the sides is not that matching. It means the complementarity of the seed sequence of the microRNA with the target is important for cleavage. So is this clear? Basically the microRNA is of 19 base pair. It is not compulsory that microRNA has to have 100% complementarity to the target site. There is a seed region in the microRNA which is a haptameric sequence in the microRNA, 7 base pair sequence in the center of the microRNA. If that region is showing the complementarity with the target site, then also it can target that particular gene. It can cleave that particular gene. Is this clear now? So here as you can see there are mismatches here also and here also, but it is showing the complementarity at the seed region. That's why it is showing the cleavage and multiplicity is 1. Sorry.
It's showing maximum of 4 bases in terms of complementarity, not 7. In which sequence? If the same sequence, maybe
First.
2 and 3. Second and third is saying that
4 base pairs? No. These are matching. That one dot is for wobble pairing, and that is also the matlab authenticated pairing.
Allowed for mismatches, no? Two mismatches and wobbles are allowed, I think.
Yes, but wobble pairing is also authenticated pairing. We can just go with the wobble pairing, and we can add that wobble pairing in the seed sequence. Is it clear or not? Any doubt? That one dot is for wobble pairing and 2 dots for complete complementarity. So these are the results of the second tab. Now you can open third tab. Here you have to submit both the files, small RNA as well as the target. So you can just choose the file or paste the sequences, anything of both microRNA and CDS new. MicroRNA in the small RNA file and CDS new in the target file. With the same parameters, just upload and submit. Have you got the results? Have you all got the results? Second row. Okay. No results? Okay. Now have you got the results now? All of you? So these are the same results, but in this we have used specific sequences of both microRNA as well as the target genes, and it is showing the base pairing, alignment, expectation, target accession, microRNA accession, that is all same, and multiplicity also. So these are the 3 tabs we can use. Now, why they have given 3 tabs?
फर्स्ट डाटाबेस को ईमेल का है और मैं ऑफलाइन जो हमारे पास आता है
Yes, so that we can we have options.
मैम एक छोटी जो सीड सीक्वेंस होती है कॉमन होती है माइक्रोआरएनए की स्पेसिफिक होती है माइक्रोआरएनए
वो हर माइक्रो आरएनए की डिफरेंट होती है. सीड सीक्वेंस इज डिफरेंट बिकॉज सीक्वेंस इज ऑल्सो डिफरेंट, सो सीड सीक्वेंस इज ऑल्सो डिफरेंट.
आइडेंटिफाई कैसे करेंगे स्ट्रक्चर से
Every microRNA has some structure, yes, from the secondary structure of the microRNA. That the main center region of the microRNA that will give the seed sequence.
हमारी एक्सपेक्ट वैल्यू मैक्सिमम ज़्यादा हो तो बेटर है
एक्सपेक्टेशन वैल्यू जो है वो जितनी कम होगी उतना ज़्यादा चांसेस हैं दैट माइक्रो आरएनए विल पेयर विद द टारगेट और सीडीएस रीजन. Expectation value, unpaired energy, these are inversely proportional. Minimum less is the unpaired energy, more are the chances. Less is the expectation value, more are the chances. So this is all about psRNA target server. So this is just input and output of the psRNA target. You can also read the paper if you want more details about psRNA target. Online paper is available easily, open access paper of psRNA target. You can just search the paper and read it.
जो ट्रांसलेशनल इंप्रेशन होता है वो ट्रांजिएंट होता है कि परमानेंट
Translational repression,
इनहिबिशन जो है
translational inhibition. What about like a transient or permanent means?
टेम्परेरी है कि मतलब वो
Like protein is not forming, then it is not forming, na? There is nothing transient in the protein formation. Protein is not forming because microRNA once bind to the target, it will repress it. So the protein is not forming because that translation is hindered at that point. Any other doubts? Anything other? So thank you for this tutorial.